Hi, in this video, we'll talk about GQ signaling, and specifically, we would talk about the role of IP3 and DAG as the second messenger in this GQ signaling pathway. It is also known as colloquially IP3 DAG pathway. So we know that in case of G protein coupled receptor signaling, there are several kind of G protein alpha subunits. One could be G alpha S stimulatory, there could be G alpha Q or G alpha I. In this particular video, we'll talk about G alpha Q. So the signaling starts when the specific ligands bind to the G protein coupled receptor signaling on the cell surface. Now, as a result, internal to the cell surface, the G protein, trimeric G protein, which is hanging out from the membrane by several isoprenoid residues, are getting activated. They dislocalize from the complex and move and diffuses along the plasma membrane to activate phospholipase C. Now, phospholipase C cleaves one specific type of lipid present in the one specific type of phospholipid present in the membrane itself, known as PIP2. Now, it cleaves it and forms IP3, inositol triphosphate, 3,4,5 trisphosphate, and diacylglycerol. Now, this IP3, which is produced, it's going to move towards the cytoplasm and diffuse in the cytoplasm. After that, it binds to the specific IP3 receptors on the endoplasmic reticulum. And once IP3 binds to the endoplasmic reticulum, it allows a specific outflux of calcium into the cytosol. Now, depending upon which phospholipase is getting activated, these membrane phospholipids could be cleaved at several residues. And here is a diagram in the inset shows where is the putative cleavage site for phospholipase C, D, or phospholipase A. Here in this video, we only talked about phospholipase C. Now, after the calcium level has risen up in the cytosol, just to give you an idea, inside the endoplasmic reticulum, calcium levels are in orders of micromolar, whereas in cytosol, the calcium level is kind of several hundreds nanomolar. So there is a huge difference between the cytoplasmic calcium concentration versus the ER calcium concentration. And a sudden rise in the calcium concentration works like a signal. So it's also a second messenger. And it is recognized by specific calcium binding protein known as calmodulin. Now, calmodulin has specific calcium binding domains known as EF hand domains, which can recognize calcium and bind to that. Now, this calcium and calmodulin complex can in turn activate several kinases such as CAMK2, CAMK4, and several other CAM kinases, all of which phosphorylate several downstream targets. Now, what are these downstream targets that these kinases phosphorylate? These targets could be very different, for ranging from regulatory elements of metabolic pathway. Let's say the CAMK2 or any CAM kinase can phosphorylate a regulatory kinase in a biosynthetic pathway, and thereby regulating the biosynthetic pathway. And thereby giving a physiological outcome. It can also work at a receptor level, phosphorylating receptor maybe, and thereby control the amount of signal transduction itself. It can also regulate a transcription factor localization. Let's say it phosphorylate one transcription factor such that it can localize inside the nucleus or might not localize inside the nucleus, and thereby indirectly controlling transcription of several other target genes. Now, other than that, it can phosphorylate several other cell cycle controlling genes, cell cycle checkpoint genes, and thereby it can have a broad variety of physiological responses. Now, we talked about IP3, which is a clipped product from PIP2, but other than that, there is something sitting in the membrane, which is DAG, diacylglycerol. Now, diacylglycerol can also bind to protein kinase C. Now, protein kinase C is a kinase, so it can activate several other downstream target by phosphorylating them. So, and thereby triggering several other metabolic responses or any other cellular responses. So, this was all about the pathway. Now, we just take a look at GS signaling and GQ signaling just to have a good comparison between that. Now, in GS signaling, what happens is G alpha subunit activates adenylate cyclase, whereas in GQ signaling, the G alpha Q subunit activates phospholipase C. Now there is activation of protein kinase A in GS signaling, whereas there is elevation of calcium level and thereby activation of calcium calmodulin kinases in case of GQ signaling. So even if several G protein, the overall framework is somewhat similar, but the G proteins involved in these pathways are different, especially at the G alpha subunit level, and thereby both these pathways have different outcome. So I hope you enjoyed this video and this video was informative for you guys. If you like my video, don't forget to like, share and subscribe. And if you have any specific questions, ask me in the comment section. Thank you.